In this lecture, we'll examine the mechanisms of electron transport and oxidative phosphorylation. These processes account for most of the ATP generated in living cells. Acetyl-CoA is the end product of lipid, carbohydrate, and protein metabolism. Acetyl-CoA is used in the TCA cycle to generate reduced nucleotides, NADH and FADH2. Those reduced nucleotides then donate their high-energy electrons into the electron transport pathway, which generates a proton gradient across the inner mitochondrial membrane. That proton gradient is used by the ATP synthase to drive the synthesis of ATP from ADP and inorganic phosphate. This is an artist's rendition of a mitochondrion. In eukaryotic cells, the electron transport and oxidative phosphorylation systems are located in the inner membrane of the mitochondria. Oxidative phosphorylation is a composite of two biochemical processes: electron transport-driven proton pumping and proton-driven ATP synthesis. These two processes work in tandem to produce ATP from reduced nucleotides such as NADH and FADH2. This simplified scheme shows how electron transport creates a proton gradient across the inner mitochondrial membrane and how the ATP synthase uses the proton gradient to drive the synthesis of ATP from ADP and inorganic phosphate. Electron transport is a process in which the transport of protons out of the mitochondrial matrix is energized by the flow of electrons through various protein complexes within the mitochondrial inner membrane. Electron transport leads to the formation of a proton gradient with a high proton concentration in the inner membrane space and a low proton concentration in the mitochondrial matrix. The ATP synthase enzyme uses the proton gradient formed by the electron transport system to drive the synthesis of ATP. Protons flow downhill from the intermembrane space through the ATP synthase protein. The downhill flow of protons from an area of high concentration in the intermembrane space to an area of low concentration in the mitochondrial matrix through the ATP synthase energizes the synthesis of ATP from ADP and inorganic phosphate. This diagram gives a more detailed breakdown of the process of oxidative phosphorylation. In the electron transport phase, reduced nucleotides pass their electrons through a series of enzyme-bound cofactors. At various stages of the electron transport system, the downhill transfer of electrons is coupled to the uphill transport of hydrogen ions. The hydrogen ions are pumped from the inner matrix of the mitochondria to the region between the inner and outer mitochondrial membranes. In the phosphorylation phase, the hydrogen ion gradient created by the oxidative proton pumps is used to drive the synthesis of ATP from ADP and inorganic phosphate. In this process, the protons flow through an ATPase down their concentration gradient back across the inner mitochondrial membrane. The respiratory chain consists of four complexes, 3 proton pumps, and a physical link to the citric acid cycle. High potential electrons from NADH enter the system at NADH-Q oxidoreductase, complex 1. Electrons flow from NADH to coenzyme Q through complex 1. The flow of electrons through complex 1 is coupled to the pumping of 4 protons out of the matrix of the mitochondrion into the space between the inner and outer mitochondrial membrane. Electrons from FADH2, which have a lower potential than those from NADH, flow to coenzyme Q through complex 2. This complex does not pump any protons. 2 electrons are carried through the mitochondrial membrane from complex 1 or complex 2 to Q cytochrome C oxidoreductase by reduced coenzyme Q. This coenzyme is lipid soluble and always stays in the membrane. 2 electrons flow from QH2 through Q cytochrome C oxidoreductase, complex 3, to the water-soluble protein cytochrome C. The flow of electrons through complex 3 is coupled to the net transport of 4 protons into the space between the inner and outer mitochondrial membrane and the uptake of 2 protons from the mitochondrial matrix. Each molecule of reduced cytochrome C carries 1 electron from Q cytochrome C oxidoreductase to cytochrome C oxidase, complex 4. 4 electrons from 4 molecules of cytochrome C flow through complex 4 to react with oxygen and form water as the final product of the respiratory chain. This process is coupled to the uptake of 4 protons from the matrix, which react with oxygen to form 2 water molecules, and the transport of 4 additional protons from the matrix to the space between the inner and outer mitochondrial membrane. There are a number of cofactors that participate in electron transport. Those are NADH, FADH2, FMN, non-heme iron-sulfur complexes, ubiquinone, and cytochromes B, C1, C, A, and A3. There's a significant difference in the absorbance spectrum of NAD+ and NADH. Such alterations in the absorption spectra occur in various cofactors depending on their oxidation states. These changes are useful in following the progress of a biochemical reaction. The first acceptor of electrons from NADH in complex 1 is FMN. The reduction of FMN to FMNH2 occurs on the same isoalloxazine ring and has essentially the same chemistry as we have previously seen with the conversion of FAD to FADH2. When the FMN cofactor is reduced to FMNH2, it accepts 2 electrons and 2 protons on the isoalloxazine ring. The protons occupy sites where there were paired electrons on 2 of the ring nitrogen atoms. There's a shift in the double bond pattern on 2 of the isoalloxazine rings when the additional electrons are added to the system. There are 3 types of iron-sulfur clusters involved in the electron transport scheme. Actually, non-heme iron proteins containing these types of clusters are found in complexes 1, 2, and 3. The least complicated variety of iron-sulfur cluster contains 1 iron atom and sulfur atoms of 4 cysteine residues. The iron ion participates in oxidation-reduction reactions by accepting or donating electrons. In the process, that iron ion can change its charge from +2 to +3. The proximity of the 4 protein sulfur atoms aids in the oxidation-reduction reaction and helps determine the energy levels of the 2 oxidation states. The next most complicated version of the iron-sulfur cluster contains 2 iron atoms, 4 cysteine sulfur atoms, and 2 inorganic sulfur atoms. The 2 inorganic sulfur atoms are not connected directly to the protein. The addition of those sulfur atoms to the protein complex involves post-translational protein modification mechanisms. As with a simpler 1 iron cluster, the proximity of the protein sulfur atoms and the inorganic sulfur atoms influences the oxidation state of the 2 iron ions and helps determine their energy levels. The most complex of the iron-sulfur clusters is a cubic structure which contains 4 iron atoms, 4 cysteine sulfur atoms, and 4 inorganic sulfur atoms. Again, the protein sulfur atoms and the inorganic sulfur atoms influence the oxidation-reduction profile of the 4 iron ions. The lipid-soluble cofactor ubiquinone, also known as coenzyme Q, can accept 2 electrons and 2 protons. Its reduction occurs in a sequential manner. The addition of 1 electron and 1 proton produces a semiquinone, a free radical with an unpaired electron. The semiquinone is a very reactive and unstable intermediate which has to be closely sequestered within the active sites of enzymes. The addition of a second electron and proton produces the stable reduced intermediate, ubiquinol. The coenzyme Q molecule carries electrons within the inner mitochondrial membrane between complex 1 and complex 3. It also functions to carry electrons between complex 2 and complex 3, and also within the cytochrome C reductase complex. There are a number of cytochrome proteins that function in the electron transport chain: cytochromes B, C1, C, A, and A3. The soluble protein cytochrome C shuttles electrons between the coenzyme Q cytochrome C reductase and cytochrome C oxidase complexes. In addition, cytochromes B and C1 are components of cytochrome C reductase, and cytochromes A and A3 are components of cytochrome C oxidase. The cytochrome proteins all contain heme prosthetic groups with a tetraperol organic structure and a central iron ion. They're covalently attached to the cytochrome protein molecules by thioether bonds to protein cysteine residues. The cytochromes differ from one another in protein sequence, the structure of the attached heme group, the absorption spectrum, reduction potential, and their role in the electron transport scheme. This slide shows the structure of the heme group of cytochrome C. The prosthetic group of cytochrome C is very similar to the heme found in hemoglobin, except that it's covalently attached to the protein through two thioether linkages. The heme side chains that are connected to the protein were unsaturated vinyl groups prior to attachment to the protein. The double bonds of these vinyl groups were reduced when the heme was attached to the protein cysteine residues. Similar to many of the molecules in the electron transport chain, cytochrome C has an absorption spectrum. It absorbs light in the visible wavelength, giving it a color which is visible to the human eye. That spectrum changes depending on whether the molecule is oxidized or reduced. Such changes in absorption are useful to biochemists because they provide a window through which investigators can follow the reaction progress as these molecules are oxidized and reduced. The cytochrome C molecule is a comparatively small protein with only 104 amino acids. That makes it relatively easy to purify and sequence. Because of its importance in biology, cytochrome C has been sequenced from many different species. The results shown here indicate that of the 104 amino acids in the protein, about 27, highlighted in yellow, are invariant in 60 widely divergent species of organisms. Another 14, highlighted in blue, are highly conserved, meaning that amino acid substitutions at those positions generally replace one amino acid with a similar amino acid. For example, leucine with isoleucine. The amino acids that are invariant are probably critical to cytochrome C function. Mutations causing changes in those amino acids would inactivate cytochrome C, and so such mutations are lethal and are not retained in the gene pool. The amino acids that are not highlighted appear to be less critical to cytochrome C function, and so a variety of changes have occurred in these amino acids. Geneticists have a field day comparing these amino acids from different species. It's possible to construct an evolutionary tree based on the number of amino acid changes from one species to another. This table compares the differences in the amino acid sequences of cytochrome C from a variety of organisms. Humans and chimps have the same sequence, whereas humans and parsnips are very different. I think that's reassuring. From amino acid composition data, biologists are able to construct a phylogenetic tree. The number of amino acid changes between species is shown on the branches of the tree. The assumption is that all living organisms originated from a common ancestor and that various species diverged from each other at different times in evolutionary history. The number of amino acid differences correspond to the passage of time since divergence occurred. This particular phylogenetic tree relates to cytochrome C, but similar trees have been constructed using other protein molecules. The fact that there is good agreement between different proteins lends strong support to evolutionary theory. We're now ready to look at some details of the complexes that are involved in electron transport. Remember that when NADH is the donor, the electrons flow from complex 1, NADH cytochrome Q reductase, to complex 3, cytochrome Q, cytochrome C reductase, and then to complex 4, cytochrome C oxidase. In complex 1, 2 electrons flow from NADH to coenzyme Q. The pathway of electron flow is NADH to FMN through a series of iron-sulfur proteins to cytochrome Q. The final product of this process is reduced coenzyme Q, QH2. The flow of electrons through complex 1 is coupled to the pumping of 4 protons out of the matrix of the mitochondrion into the space between the inner and outer mitochondrial membrane. 2 additional protons are taken up from the matrix to convert coenzyme Q to QH2. The second complex in electron transport is cytochrome C reductase. The QH2 that's produced in complex 1 diffuses through the hydrophobic membrane to complex 3. There's an interesting transition in this complex. That is, QH2 feeds 2 electrons into the complex, but cytochrome C only accepts 1 electron. As a result, it takes 2 cycles of reduction involving 2 cytochrome C molecules to effectively convert coenzyme Q from the reduced to the oxidized form. In the first cycle, part A, the reduced coenzyme Q donates 2 electrons. 1 electron goes through the iron-sulfur protein and a protein-bound cytochrome C1 to cytochrome C. The other electron goes to a protein-bound coenzyme Q molecule to form a free radical, then through 2 variants of cytochrome B, and finally ends up in a second coenzyme Q as a free radical with an unpaired electron. This free radical intermediate is extremely reactive and could damage the cell if it were set free from the surface of the enzyme. Fortunately, the enzyme complex holds the intermediate closely until further reaction can occur. In the next cycle, part B, a second molecule of reduced coenzyme Q donates 2 electrons. 1 electron follows the same pathway as in the first cycle through an iron-sulfur protein and a protein-bound cytochrome C1 to cytochrome C. The second electron follows the pathway used by the second electron in the first cycle and ends up converting the free radical back to reduced cytochrome Q. The result of this elaborate electronic dance is that 2 molecules of reduced coenzyme Q donate 4 electrons. 2 of those electrons produce 2 molecules of reduced cytochrome C, and the other 2 electrons end up regenerating a molecule of reduced coenzyme Q, QH2. If you thought that the last reaction sequence was complicated, here's something even more baroque. The figure summarizes the last set of reactions in the electron transport chain, which is catalyzed by cytochrome C oxidase. Keep in mind that all 4 electrons enter this reaction scheme provided by reduced cytochrome C. The end result of this series of reactions is the reduction of molecular oxygen, O2, to 2 molecules of water. It takes 2 electrons to reduce each oxygen atom to water, so 4 electrons are required. We'll start at the upper left with a complex having an iron ion in the +3 state and a copper ion in the +2 state. The addition of 1 electron from cytochrome C reduces the copper ion to the +1 state. The addition of a second electron reduces the iron ion to the +2 state. At that point, molecular oxygen binds to one of the ligand binding sites on the iron ion. Then an internal oxidation-reduction reaction takes place. Both the iron and copper ions lose an electron, and the molecular oxygen is converted to a double anion. In the next step, an electron and 2 protons are added, and molecular oxygen is split into 2 unconnected oxygen atoms. One of the oxygen atoms, which is attached to copper +2, gains 2 protons and 2 electrons to form water. The other oxygen atom, which is connected to a really weird iron +4, ends up carrying a -2 charge. The addition of 1 more electron and 2 protons produce a second water molecule and return the iron ion to the +3 state. At this point, the cycle is complete. This slide summarizes the fate of protons in the cytochrome C oxidase reaction. During the process of converting molecular oxygen to water, 4 electrons from reduced cytochrome C and 4 protons from the matrix are used to reduce the 2 oxygen atoms to water. In addition to the 4 chemical protons absorbed from the matrix and incorporated into water, 4 additional protons are pumped from the matrix into the intermembrane space. Here's a more detailed diagram showing not only the 4 complexes that participate in electron transport, but also some of the key participants in those complexes. We'll now begin a discussion of the standard reduction potentials of these component reactions. Remember that the electron transport process starts with NADH donating electrons to the system and finishes with oxygen receiving electrons to form water. The critical point is that as the electrons pass through the various carriers, there's a gradual loss of reducing potential and a corresponding release of energy. Some of that energy is used to pump protons across the inner mitochondrial membrane to create the proton gradient. This is the Nernst equation, which has been beloved by physiologists for a number of generations. The Nernst equation is similar to the free energy equation in that it allows us to calculate the potential energy of chemical reactions. However, the Nernst equation is used for oxidation-reduction reactions, and the energy values are expressed in volts, not in joules or calories. Take note that this is a biologist's formulation of the Nernst equation with the hydrogen ion concentration set at pH 7 to reflect the normal condition that prevails in living cells. The Nernst equation tells us that the reduction potential for any reaction is equal to the standard state reduction potential plus RT over NF natural log of the electron acceptor over the electron donor. This slide gives a generalized equation for an oxidation-reduction reaction. The standard state voltage change, delta E sub 0 prime, for the combined reaction equals the standard reduction potential for the electron acceptor, the substance being reduced, minus the standard reduction potential for the electron donor, the substance being oxidized. The slide also gives a formula for determining the standard state free energy change for reaction if you know the value of the voltage change. That formula is delta G 0 prime equals minus NF delta E sub 0 prime. This formula allows us to compare standard state voltage change in a reaction with the standard state free energy change. In other words, we can convert voltage changes to joules or calories. Note that there's a minus sign on the right side of the equation. That means that an energy-releasing reaction, which has a negative delta G 0 prime, will have a positive delta E sub 0 prime. This is a table showing the standard reduction potentials for a number of the participants in the electron transport pathway. These are half-reaction potentials which are measured against an arbitrary standard, a hydrogen electrode. The reactions are shown in the direction of reduction. In order to correspond to the real world, there must be another half reaction involved, which would be going in the direction of oxidation and which would serve as a source of electrons for the reduction half reaction. This table was constructed using a hydrogen electrode as the other half reaction, but one can combine any 2 half reactions to determine the voltage change for a physiologically relevant oxidation-reduction pair. If you combine 2 half reactions from a table such as this, one of the reactions runs in the direction of reduction and uses the E sub 0 prime value shown in the table. The other half reaction runs in the direction of oxidation, reversed from that shown, and the E sub 0 prime value is given the opposite sign from that shown in the table. For example, a positive value becomes a negative value of the same magnitude. Here's a relevant example of how to calculate the change in standard state reduction potential for an oxidation-reduction reaction. This particular example corresponds to the change in standard state reduction potential for the overall electron transport process, starting with NADH donating electrons and ending with molecular oxygen being converted to water. The upper 2 equations show the half reactions and give the standard reduction potentials for the reduction of NAD plus to NADH and the reduction of oxygen to water. However, because NADH is being oxidized in the electron transport process, we have to reverse the direction of that half reaction and change the sign of its E sub 0 prime. The two reactions shown on the lower part of the slide can then be added to give the overall reaction and the net voltage change, delta E 0 prime. The net voltage change for NADH oxidation by the electron transport system is plus 1.14 volts. This is a very revealing diagram. The reduction potentials of the electron transport participants are plotted in the order that they occur in the overall pathway. Notice that as the sequence proceeds, there's a constant drop in reduction potential. There's a continuous loss of energy. In addition, there is more than sufficient drop in voltage in each of the complexes 1, 3, and 4 to account for the synthesis of one molecule of ATP. In fact, if the process were 100% efficient, there'd be enough energy released by each complex to synthesize at least 2 molecules of ATP. Complex 1, which accepts electrons from NADH, passes them through FMN and donates them to coenzyme Q. It has a voltage drop of about 0.4 volts. Complex 3, which starts with coenzyme Q and ends with cytochrome C, has a drop of about 0.3 volts. Complex 4, which accepts electrons from cytochrome C and donates them to molecular oxygen, has a drop of about 0.45 volts. We'll take a second look at this simplified scheme to reflect on how the energy from electron transport is used to create a proton gradient across the inner mitochondrial membrane, and then how the ATP synthase captures the energy from that proton gradient to drive the synthesis of ATP from ADP and inorganic phosphate. Notice that the role of the electron transport complexes is to pump protons across the membrane. These complexes are not directly linked to the ATP synthase, and they're not directly involved in ATP synthesis. The connection between electron transport and oxidative phosphorylation is through the proton gradient. The synthesis of ATP is catalyzed by the ATP synthase. The ATP synthase complex is made up of an F0 component, which is embedded in the inner mitochondrial membrane, and the F1 component, which is a peripheral protein assembly. The F0 component consists of 2 different protein subunits, and the F1 component contains 5 different subunits. Here's some experimental evidence for the involvement of the F1 component in ATP synthesis. In this slide, we see a series of 4 electron micrographs of mitochondrial preparations. Panel A shows peripheral proteins, the F1 part of the ATP synthase, lining the matrix surface of the inner mitochondrial membrane. In panel B, those proteins have been removed from the mitochondrial preparations by treatment with urea. Panel C shows the isolated F1 components of the ATP synthase. In panel D, those F1 components have been added back to the mitochondrial fractions. It turns out that mitochondrial membrane preparations that contain the F1 component can synthesize ATP from ADP and inorganic phosphate as long as the membrane structure remains intact and there's a gradient of hydrogen ions. When the F1 component of the ATP synthase is removed, the ability to synthesize ATP is lost. When the F1 component is added back, the reconstituted preparation regains the ability to synthesize ATP. The F1 complex has 3 alpha and 3 beta subunits surrounding a central gamma subunit. The gamma subunit passes through the middle of the alpha 3 beta 3 hexamer, which consists of alternating alpha and beta subunits. At any given time, each of the 3 beta subunits exists in a different nucleotide binding form, designated O, L, and T. These 3 forms are interconvertible. The alpha subunits bind ATP that do not participate in any catalytic or transport reaction. Proton-driven ATP synthesis involves a binding change mechanism in which 3 sequential 120-degree rotations of the gamma subunit drives the beta subunits through the 3 different forms, T tight, O open, and L loose. The subunit in the T form converts ADP and inorganic phosphate to ATP, but does not allow the ATP product to be released. When the gamma subunit is rotated by 120 degrees in a counterclockwise direction, the T form is converted into the O form, allowing ATP release. Then ADP and inorganic phosphate can bind to the O form. An additional 120-degree rotation traps ADP and inorganic phosphate in the L form. This figure shows an actin filament bound to the gamma subunit. This is an artificial hybrid system constructed in a research lab to demonstrate that the gamma subunit actually rotates. In the experiment, the hydrolysis of ATP drives the rotation of the gamma subunit. This could be directly observed by fluorescent microscopy. Under physiological conditions, there would be no actin present, and the rotation of the gamma subunit would drive the synthesis of ATP from ADP and inorganic phosphate. The membrane-spanning F0 component consists of A and C subunits. The C subunit consists of two alpha helix structures with a negatively charged aspartate in the center. The A subunit consists of a cytoplasmic and a matrix half-channel. Each of the C subunits consists of two alpha helices. Between 10 and 14 of the C subunits form a membrane-spanning ring. An aspartic acid residue in one of the helices lies at the center of the membrane. These aspartate residues are protonated and deprotonated during the passage of protons around the ring. The A subunit appears to include two half-channels that allow protons to enter and pass part way, but not completely, through the membrane. Proton movement across the membrane drives rotation of the C ring. A proton enters from the intermembrane space into the cytoplasmic half-channel of the A subunit to neutralize the charge on an aspartate residue in a C subunit. With this charge neutralized, the C ring can rotate clockwise by one C subunit. This moves another protonated aspartic acid residue out of the membrane and into contact with the matrix half-channel. This proton can diffuse into the matrix, resetting the system to its initial state. In summary, each proton enters the cytoplasmic half-channel, follows a complete rotation of the C ring, and exits through the matrix half-channel. The flux of protons through the F0 component drives the rotation of the C ring, which in turn drives the rotation of the gamma subunit of the F1 component. The rotation of the gamma subunit in turn powers the synthesis of ATP from ADP and inorganic phosphate. This overall process constitutes the phosphorylation part of oxidative phosphorylation. When we calculate the ATP yield from various catabolic pathways, there's one additional factor we have to take into account. That's the energy cost of transporting materials from one site to another within the cell. An excellent example of this energy cost is the metabolic fate of the NADH produced by glyceraldehyde 3-phosphate dehydrogenase. That NADH is produced in the glycolysis pathway, which is located in the cytoplasm of the cell. However, the NADH is used by the electron transport system in the mitochondrial inner membrane. There's a metabolic system called the glycerol 3-phosphate shuttle that accomplishes the translocation of NADH from the cytoplasm to the mitochondria. The reducing equivalents from cytoplasmic NADH are carried into the matrix by the glycerol 3-phosphate shuttle. The end product of this process is FADH2 rather than NADH. The net cost of this transport is that one ATP equivalent is lost during the process because FADH2 only yields 1.5 ATPs in the oxidative phosphorylation system. In the glycerol 3-phosphate shuttle, electrons from NADH are used to reduce dihydroxyacetone phosphate to glycerol 3-phosphate. The glycerol 3-phosphate is reoxidized by electron transfer to an FAD prosthetic group of a dehydrogenase that's located in the inner mitochondrial membrane. Subsequently, the electrons are transferred to coenzyme Q, forming QH2, which enters the electron transport chain. This mechanism is predominant in muscle cells. In the heart and liver, electrons are carried into the matrix by the malate aspartate shuttle. This convoluted mechanism is mediated by 2 membrane transport systems and 4 enzymes. The major benefit of this elaborate process is that the electrons from cytoplasmic NADH end up in mitochondrial NADH, so there's no reducing power lost in the process. The malate aspartate shuttle begins with the reduction of oxaloacetate to malate. The shuttle process involves 2 antiporters. One antiporter exchanges cytoplasmic malate for alpha-ketoglutarate, and the other exchanges aspartate for glutamate. The overall reaction scheme involves the sequential formation of malate, oxaloacetate, a transamination reaction to form alpha-ketoglutarate and aspartate, and a second transamination reaction to form glutamate and oxaloacetate. Earlier biochemical studies suggested that 3 ATPs were generated from NADH, whereas 2 ATPs could be derived from FADH2. However, recently this stoichiometry has been revised to 2.5 ATPs for NADH and 1.5 for FADH2. The following energy yields are seen in oxidative phosphorylation. Electrons that enter the electron transport system from NADH yield 2.5 ATP. Electrons that enter electron transport system from FADH2 yield 1.5 ATP. Electrons from NADH pass through complex 1, which pumps protons, and then go on through complexes 3 and 4, which also pump protons. Electrons from FADH2 pass through complex 2, which does not pump protons, and then go on through complexes 3 and 4, which do pump protons. As a result, there's less of a proton gradient produced from FADH2, and thus less ATP is produced. So here's the latest up-to-the-date scoop on ATP production from the complete oxidation of one glucose molecule. We take into account the latest estimate of ATP yield per NADH and FADH2 and the energy cost of moving reducing power from the cytoplasm into the mitochondria. Using these values for ATP production, the complete oxidation of one glucose molecule should yield about 30 ATPs. Some organisms have a mechanism for using the electron transport system to generate heat rather than ATP. An uncoupling protein, UCP1, acts as a proton channel that allows protons to enter the matrix of the mitochondria without passing through the ATP synthase. This mechanism dissipates the proton gradient and generates heat, but eliminates the synthesis of ATP. In addition to natural systems for uncoupling oxidative phosphorylation by dissipating the proton gradient, there are also natural and chemical substances which work directly to inhibit the flow of electrons through the electron transport system. There are a variety of such inhibitors that exhibit specific inhibition at each of the electron transport complexes. In the presence of these inhibitors, no NADH is oxidized. The formation of ATP by oxidative phosphorylation can also be prevented by chemical uncouplers. For example, 2,4-dinitrophenol uncouples oxidative phosphorylation. Reagents such as 2,4-dinitrophenol act like the protein UCP1 in that they allow electron transport to proceed in a normal manner, but they dissipate the proton gradient by allowing protons to leak back into the matrix. This person is getting ready to launch a kayak. There are many wonderful sights to explore on the Washington coast. The river that runs through Ruby Beach is easy to ford during the dry season, but in rainy weather crossing can be very challenging. Because of all the logging activity going on nearby, there's always a lot of interesting driftwood on these beaches. This young woman is keeping her toes dry by standing on a rock. It looks so interesting on the other side of the river, but do I really want to get my hiking shoes totally soaked? When you see a sky like this, you never know whether it will be sunshine or rain. It's always a good idea to have a waterproof jacket along. It was dry when this photograph was taken, but you can see the slanting rain falling in the distance. I love the word flotsam, but why? I don't know. This pile of flotsam probably came from some fisherman's crab pot. The quality of photographs is dependent on light. On this day there was a partially cloudy sky, but at ground level there was some persistent fog. Perfection. These holy rocks are so interesting. Think how hard some sea creatures had to work to burrow out these rocks. I think this would make a good cover photo for a mystery story. Who did it to who on Hoo-Hoo Beach? Where are these folks coming from? When they disappear into the mist, where will they be going? The shell is there, but the crab is long gone. On this day the river is shrouded in fog as it flows slowly into the sea. How many years of erosion did it take to produce this mushroom-shaped rock? This photograph wouldn't be that much without the tiny figures in the background. Well, maybe it isn't that much anyway, but I like it. How did that guy get up on top of this sea stack? If I tried that, the result would be a broken neck.